Hey, how are you?
I'm good. How are you?
I'm good. How's your sleep and all that stuff?
My sleep? It's been okay. Yeah, I mean, yeah, it's normal till now.
Uh-huh.
Hopefully it stays good at my I can't like sleep in. My body just wakes up because I've been doing it for so long. Yeah.
Are you like able to sleep like at night? Because I know we talked about um, having trouble falling asleep.
Yeah. Yeah, I mean I feel like I've been sleeping at least like 7 hours. That's good.
Uh-huh. So it's like you remember how we talked about like, you know, okay. So
Yeah.
all right. Yeah, awesome. Okay. Yeah. What else have you what have you been working on? Just like Anki and stuff?
Yeah, that I've really just been like doing as much like Soshanki as possible today.
Yep.
Yeah, I mean there's so many like I don't think I'm ever gonna be able to finish it, but yeah, it's good to review some concepts. So I'm just trying to do as much as I can
Got
today
it.
and
Got
tomorrow.
it. Okay. Yeah. What would you like to go over today?
Um, since I've only been doing those cards, I don't really have something specific.
Mhm.
Um, I guess maybe I can think of like
Yeah,
uh,
bring
uh
like the about because you like did the section bank again for like BB and CP, right?
I started CP, but I didn't finish it.
Got it.
Yeah, I think yesterday when we spoke, like I said, I didn't do it. Like I did do it last year, so like I just haven't like redone CP, if that makes any sense.
Yeah, yeah, of course. What about like the psych soc? Like, how do you feel about that section bank part?
Yeah, I haven't redone it, but I feel like the best way for me right now with Psych Soc is just to familiarize myself with as many concepts as possible. Because I feel like with the experimental part of it, I think I have a good handle of it with Psych Soc.
Got it. Okay, got it. So, okay, let's see what we can work on.
I'm thinking, I think that, well, I guess one thing for the formula sheet, because I was also reviewing some of my formulas today, that we talked about, and I think I am leaning towards like writing down like the equations for like the kinematic equations
Mhm.
and like the electromagnetism equations first thing. I still have to like practice writing it down and timing myself, but yeah, I think I switched to like doing that because I think those I feel less comfortable about. So.
Got it. Got it. Okay. Um, so let's see. Um, so okay, so you've been mostly going over psych soc cards. Um,
There's
yeah,
a topic which I just also always just
yeah.
I don't know, I just don't distinguish much between things there, which is like the DNA repair mechanisms.
Uh-huh.
I know that there's different types, and I know like exonuclease and endonuclease. But like the specific ones, like uh base repairs, like the specific kinds, I'm not too familiar with.
Mhm.
I don't, yeah.
Yeah, so you can have like yeah, DNA polymerase um can repair some so okay, so you can have like a mismatch when you so like okay, this is like through like replication when the DNA makes more of itself. So a mismatch, if there's a mismatch in the newly synthesized DNA, like you know, like guanine attaching to like thymine or something like that. Um, what happens will be that that new DNA is going to be like that strand with that mispaired nucleotide is going to be removed, and then that missing like patch is going to be uh replaced with the correct nucleotides by a DNA polymerase.
Mhm.
So and when the DNA strand is cut, you're actually cutting like the phosphate backbone as well. So once you put in the new DNA, the once you replace that missing that patch that you got rid of, and you replace it with, the new nucleotides, there will still be gaps, and so DNA ligase will be, sealing that gap. Um, so let's see. Um, okay, so another thing that can happen would be something like um so I guess like okay, like UV light, ultraviolet light, like once it hits our skin, it can cause um something called thymine dimers, where two thymines like bond to one another, like when they're right next to each other. So you want to get rid of one of them, and so what can be done is like that strand can be like opened up, and then enzymes can cut that damaged region out, and then a DNA polymerase can replace it the same way that the previous example would do, and ligase will seal the backbone back together. So that's another type of repair. Um, and there could also be so the nucleotide excision repair. A nucleotide includes the ribose sugar and the phosphate, right? So, but you can also have a base excision repair, which is just removing that base. You know what I mean? Like if it's a uracil, we remove the uracil, but we don't, break the backbone. Yep.
Oh, I see. So that's nucleotide excision repair versus base excision repair.
Yep.
Okay.
Yeah. Here, I'm gonna look. I know I wrote a couple of these in my notes here. Let me see. Okay, I can't find it in here, but I can just look it up then.
Yeah.
Different types, but um, you can have okay. So yeah, the base excision repair, nucleotide excision repair. Um, you can have like homologous recombination. Um, so yeah, let me see what I can pull up here. Okay. All right, there seems to be some. All right, let's try this. So I'm gonna send this link in the chat, and you can open it up and
Mm.
share your screen.
Okay.
Yeah. So here we have. Okay, so let's see. Yeah, okay. Okay, if you have a mutation, it can be repaired through mismatch repair, chemical reversal, excision repair, and double-stranded break repair. Um, and so the first one they talk about is the mismatch repair, and that means that so like in this image here, we see that the mismatch, or here, let me connect my. So we see that the mismatch is that G, that guanine over there, right?
Yeah.
So yeah, so we have our guanine here, which is mismatched with the thymine in the template strand. And um and so what happens is that that new DNA strand is cut. It's cut like out. Um and like an enzyme would do that. So okay, I guess I should say that the DNA polymerase like will like kind of like proofread as it goes through like a DNA strand. And something will remove it. DNA polymerase will add nucleotides to fill in what's removed, and then the ligase is just going to seal the gap. So yeah, so we see that for this, they're cutting like here and like here to get this guy out. And then we have our like the gap in that next the image that goes below that. So that's gone. And then if you go down a little bit more, we see that the missing patch is replaced by with correct nucleotides by a DNA polymerase. Um, and from here we can see that this would be a 3' and this would be a 5', right?
Yeah, from that strand and from this strand, yeah.
Uh-huh. And when you're synthesizing, DNA, you need that 3' end, right? So that's why when the polymerase can add to that end over there. So that's why when you see this replacement DNA, you see this like they're connected, right?
Yeah.
Because it's going, from that. But on the right, it's not connected because the end of this should be a 3 and the beginning of this should be a 5, right? So what's done there is that that gap gets sealed up by ligase, just like you would see in DNA replication for like the lagging strand, right? So that's the okay, so that's just the mismatch repair. And then we can have so yeah, and then DNA damage can occur um not just during replication, but certain things in the environment like UV light, chemicals, X-rays, things like that. So, so in some cases, the cell can fix DNA damage by reversing the chemical reaction that caused it. So in this image here, we see guanine. And if it moves to the right, it has a reaction with a harmful, like when it has a reaction with a harmful chemical, the guanine gets methylated. So we have a methyl group over there. So that's, through a chemical reaction. But if you add in an enzyme that removes the methyl group, it will turn that methylated guanine back into regular guanine.
Mhm.
So that's just an example of repairing DNA damage by reversing the chemical reaction that caused it. So okay, that's that type of repair. And then we have nucleotide excision repair, which will so yeah, the base
Please.
and the so okay, so this is the phosphate here. This is the, base, right? So okay, so the base plus the ribose sugar would be called a nucleoside. But the base with the ribose sugar and the phosphate is the nucleotide.
Yeah.
So okay, so for this, that would mean that you're going to need to cut the okay, I guess before I talk about that. So okay, the nucleotide excision repair means that it can remove and replace damaged nucleotides that warp or bend the DNA helix. So UV radiation is a good example where cytosine and thymine bases can react and form bonds that mess up the double helix, like from being a smooth double helix. And so the most common type would be a thymine dimer, which is just when two thymine bases are attached to one another. And we see that here where, we got that, right? And, unlike the two A's underneath, which are not connected to one another, the two T's on that new strand are connected to one another, right?
Yeah.
And if they're connected to one another, that means that the strand is kind of bunched up in that region, right? So that's a thymine dimer, and to repair it, you need to, well, open up. So that opening up to form a bubble, that's like when you have helicase unravel the two strands, enabling that the replication bubble. I think that's what it's called.
Yeah.
You know what I mean? Um, so okay, so that's why they're, spread apart, and then the enzymes cut the damaged region out of the bubble, and then DNA polymerase will replace the one that they cut out with the proper nucleotides, and then the ligase again seals the backbone. Okay, and then base excision repair, detect and remove certain types of damaged bases. A group of enzymes called glycosylases play a key role in base excision repair. Each glycosylase detects and removes a specific kind of damaged base. For example, a chemical reaction called deamination can convert a cytosine base into uracil, which is typically found only in RNA, as shown below. Glycosylase can remove the incorrect uracil base, and again, the DNA polymerase fills the gap. DNA ligase seals the gap. So yeah, in this picture we have, we have this uracil here, which we don't want. So what we can do is we can convert. Oh, I guess it already happened. So it used to be a cytosine there, where that U, the red U is, but deamination converted it into uracil. So we have uracil. And that doesn't really, it's not really like complementary to uh to 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 G, right? Um, and so that's something that that you would want to fix. So the uracil gets detected and removed, um, leaving leaving just the base. Oh, sorry, leaving a baseless nucleotide. So it would be just this portion.
I see.
So we're not cutting the backbone out, right? We're just changing the base. So yeah, the uracil is removed, and then a baseless nucleotide. Okay, so we first have the baseless nucleotide, and then we remove that, leaving a hole, and we do break the backbone in that case. But then it's again filled with DNA polymerase and sealed by ligase. Okay. Then the most serious type of DNA damage is a double-strand DNA break, which can cause larger regions of chromosomes to be lost if the break isn't repaired. Um, there are two pathways involved in double-stranded break repair. There's non-homologous end joining, and there's homologous recombination. So in non-homologous end joining, the two broken ends of the chromosome are glued back together. Uh, and this is messy because it involves usually the loss or sometimes the gain of some nucleotides at the site that is cut. And that means that it can introduce a small mutation, but it's better than the entire region of a chromosome being lost. So we see here some high energy radiation causes the double strand break here, and the chromosome is glued back together with but with still some type of mutation at the site at which it broke. So that's that. And then in homologous recombination, information from the homologous chromosome that matches the damaged one, or from a sister chromatid if the DNA has been copied, is used to repair the break. In this process, two homologous chromosomes come together, and the undamaged region of the homolog or chromatid is used as a template to replace the damaged region of the broken chromosome. And it's cleaner because it doesn't involve mutations. So we see that we have the same high energy radiation causing a double strand break, but instead of just gluing it together, we use a homologous chromosome that's complementary to this to identify like what the correct bases should be, and then we can seal it back together. So it's not going to have the messed up kind of mutations because you can check it with the homologous strands or the homologous chromosome.
Yeah.
Yep.
This one was, this was just a general mismatch repair. Okay, and this was nucleotide excision. So this was just like base excision. Mismatch repair is base excision, base repair. Maybe.
Sorry, what were you asking?
The first one, mismatch repair. Is it like a base repair? It's
Well, you are getting rid of that backbone right away. Like you're cutting into that backbone right away, right?
okay, so the backbone is not cut.
Yeah, I think if you go all the way down, they kind of summarize it. So yeah, so key point. So when replication mistakes are not corrected, they may result in mutations. Mutations can also arise from external sources like UV radiation. Mutation can also be benign, uh, but can also have serious consequences like cancer. All right, mismatch repair. That's what happens immediately after DNA replication. And enzymes involved in this type of repair recognize, aka like when they proofread, um, incorrectly incorporated bases, they remove them and then they from the DNA and replace them with the correct bases. And that's cutting that entire mismatch region out, like it's cutting into the strand, the phosphate backbone. Um, So that's your mismatch repair. Nucleotide excision repair. One or more damaged nucleotides in the small surrounding DNA region are removed and replaced. So that's also going to be involving, cutting the strand, like the breaking like the single strand at least. And then in base excision repair, that's where it's just the incorrect base removed and then filled in instead of like a whole new nucleotide. Double-stranded DNA breaks are the most serious type, and there's they could be repaired by either non-homologous end joining, which isn't the best because there's still going to be some type of small mutation, or homologous recombination, which uses a homologous chromosome as a template for the repair, which means that there's going to be it's more clean and there's going to be uh fewer mutations involved. And then under that, we can see more specific. So mismatch repair, this is something to fix DNA damage like putting in the wrong, inserting the wrong nucleotide, um, deleting one, or misincorporation of bases that can arise during DNA replication and recombination. So I think that just means maybe something related to, yeah, I'm not sure about that part, but I guess that would be like a substitution perhaps. Because then we would have insertion, deletion, and substitution, right? But okay, let's see. And then chemical reversal, DNA repair mechanism based on the reversal of chemical reactions that induce DNA damage, nucleotide excision repair. So that can fix something like thymine dimers. Base excision repair simply removes that base, not the entire nucleotide. Double-stranded break repair, DNA polymerase. That's the thing that can. Oh that's the thing with the proofreading and the exonuclease activity, right? So it can proofread and also cut out regions, I guess, at the ends of nucleotides to to repair them. Ligase just brings, connects the nucleotides back together. Glycosylases, I mean, I guess they're just saying mediate space excision repair. But yeah, those are the different types of repair. And yeah, what are your thoughts on that?
No thoughts, but
Huh?
yeah,
Yeah, I'm trying to find in my
just a lot of content to memorize, like the differences. Yeah.
uh-huh.
Mhm.
So okay, let me try to find some stuff on this.
Um,
Hmm?
yeah, it's something, it's unrelated.
Wait, what was that?
It's just an unrelated question that I have.
Yep, what is it?
I just want to make sure. I think I'm blanking on it. So for the timing strategies, it's like when I'm at question 20 or question 30 that I have to be at 1 hour.
Oh yeah, so I mean, yeah, if you are, if there's 60 questions, like 59 questions, right? Then and you want to break it up into thirds, right? So when you're on question 20, you should look at the clock and uh that's so it's 90 minutes, right? So or an hour and a half. So once
30.
the first third, yeah. So once you're on question 20, you want to see uh if you have 1 hour left, you're good. If you have more than an hour left, you're better than good. If you have less than an hour left, just gently tell yourself to go a little faster. And then it'll be like it can be like it doesn't have to be the next third. It can be the halfway point. So when you're on question like 30, you should have like 45 minutes left, right?
Mhm.
And then when you're on question 40, you should have the 30 minutes left, right?
Yeah, this makes sense. I don't know. I was just blanking on it right now. Maybe it's
Yeah. So,
um
mhm.
yeah, I'm nervous.
Oh yeah, of course. But you've put in your work here. So I mean, yeah, it's also natural to feel that way, of course. Um, but yeah, let me see if I'm trying to find like maybe like a passage that talks about something like that. So while I look for this, are there any other topics like that that you feel?
Um, I it's I want to review just the basic mechanisms of like E1, E2, SN1, SN2. Um, just because Oracle is not my strength. Um,
Yeah, I have in that PDF folder like a generalized kind of thing for the or like way to go over the SN1, SN2, and all that stuff.
yeah.
You can take a look at that, I think. Let's see. Oh, okay, let's try this. Um, Putting it in the chat.
Yeah, it's just taking a while to load. Okay.
Okay. All right, let's see. Yep. So why don't you try this one?
The 50th codon in an mRNA segment is mutated to the sequence TAG. What is the effect of this mutation? Well, I know like this would be what like UAG, that's a stop codon,
Mhm.
right? So it's going to stop and it's gonna have, I believe, this.
Excellent.
Which enzyme prevents the effects of mutations by proofreading DNA? It's DNA polymerase. 'cause it has exonuclease activity.
What's the yeah, all right, it's okay. Yeah, we can go on.
Which of the following mutations describe the pathology of sickle cell anemia in which an adenine is converted into a thymine? It's gonna be a point mutation.
Mhm.
I think so. Yeah. A mutation in a bird species leads to a change in the shape of the beak of all the birds of the species over a long period of time. Which of the following types of mutation is this most likely to be? It's probably going to be a beneficial mutation.
What?
Why? Because of like selection, natural selection. So it's changing over a long period of time. So like, it means like it's being selected for, not against.
So I think what will give that away is just how it says, because sure, it could take a long time for one bird species to have changes in the shape of its beak, but it says all of the birds of the species, right? So that's kind of like a founder effect kind of thing where this mutation is definitely beneficial if it led to all the other birds having it, right?
Yes.
Okay. Yep.
I've never heard of a somatic mutation. Let's see what it is. Does
What
it
do you think it
say?
would be?
Like a somatic symptom is what I'm thinking, like a physical thing, physical mutation. I'm not sure
Okay.
that is. Uh, okay. A mutation which leads to defunct ribosomal RNAs would most likely affect which of the following processes? Translation. Yeah.
Yeah. Why?
Because you need the ribosomes they're like the enzymatically active
Hmm.
ribosomes.
Made out of?
Sorry?
What are they made out of?
RNA, but
And like
something else.
some small amount of proteins as well. Where are they made?
In the nucleolus.
Excellent. Good job. Okay.
A nonsense point mutation resulted in a premature stop codon for the gene encoding for helicase, which of the following phenomena will not be observed. The mRNA transcript could include the codon. The resultant protein product will travel faster. DNA replication will be negatively affected. I think it would be negatively affected. Wait, I think so. Just because helicase opens up, allows for replication.
Well,
Oops.
so um
Oh, it's nonsense.
so
Uh, uh,
this is asking, what is the question stem asking?
which of the following will not be observed?
Mhm. Not, right?
Yeah. Oh, not.
Yeah.
Oh my gosh.
Yeah, yeah.
Yeah,
Hmm.
tricky. Yeah. Oh, I kind of messed it up because I only saw the answer.
So
Um,
so okay, yeah. Helicase is not getting made, so it's not going to be able to unravel the thing, right? So um I mean, DNA replication will be negatively affected, but we want to see what won't be, right?
yeah.
And if all it is doing is making a premature stop codon for helicase, uh, what it would not do. Well, yeah. What?
Yeah.
Yeah.
I see. Like, I mean, I can start like if
Mhm.
it's inserting a stop codon, like it could include these. And because it's
Codons,
sorry?
right? Yeah, because these are the stop codons, right?
Yeah. Um, if it's shorter, like it will travel faster through gel electrophoresis.
Exactly. Yep.
And it will be negatively affected, like DNA replication. So the only one would be um like anything prior to it, which shouldn't be affected.
Good. Good. Good.
Oh, a woman who was exposed to UV rays developed a mutation that led to skin cancer, like a thine. We just read it, the dimer, probably. I don't know. If the woman wears a child, what is the likelihood that the child will be carrier for this mutation? I think 0%.
Why?
Because it was a mutation in uh like not like autosomal like chromosomes, and uh like I don't think like plus she's a woman, so all of her like oocytes would have already been gone through like until what is it, they'd be arrested in prophase one even, so like that's also another reason why this wouldn't be the case. Um
But
yeah,
what if it's a male?
sorry.
What if it's a male?
Still, because it's I think it's still not like a mutation in like um it's like in the like autosomal chromosomes,
Uh-huh.
and and um it's like
So this is kind of like what a somatic thing would be, right?
yeah, yeah, that's what I wanted.
And I guess we can bring it back to that other choice that was talking about somatic mutation. But yeah, uh mutations that happen in cells that are not gametes like won't be passed on, right?
Yeah.
So yep, good.
What is the effect of cytosine deamination on the 5' terminal cytosine of the following DNA sequence? Deamination cytosine.
Hmm.
I'm thinking it's like one of those of the repair things that we saw that are like by like an enzyme, as I think we saw like carboxylation, I'm not sure, methylation. Um, so if cytosine, I suck at this, cytosine would be deaminated. So like that's obviously going to have an effect.
Hmm.
So I don't think it would be A. I don't think it would be D either, just because like just by like a what like a deamination change like a group. I don't think it can convert like a two ring thing to a one ring thing.
Okay.
Um there's
Do you know like what the purines and like the structures of the purines and pyrimidines?
I've been trying to remember.
Yeah,
Um
that might be a good thing to.
yeah, I think I have it open in one of these tabs even.
Yeah, so that could be a thing to brain dump on the sheet, on the scrap sheet that you get, because I'm fairly certain that it's fair game on the MCAT, the structures.
Yeah.
Um, so yeah, I would think of something like that. So like for instance, um uh let's see, it's something like this. Um and I think it's like that. Um Cytosine, yeah, so that's cytosine. Um, and it can help to remember, like, how um so you know what? So if you look at if you so that repair mechanism thing, I wanted to ask you this. Yeah, here, like what does that mean? And what does that mean?
Oh, I was thinking something along those lines with this.
Mhm.
Just because I know that the way that DNA polymerase is sensing is by like the hydrogen bonds that you would expect
Uh-huh.
on like of G and C, you would expect like three hydrogen bonds, and for the others you would expect two hydrogen bonds, and so that's the way it's sensing. So that's sort of what I was thinking.
Yep.
Yeah,
Yep.
and that would make B correct, I think. Maybe. Yeah.
This
Huh?
for this or something else? Oh, I just wanted to. So the reason I brought it up is because if you, so what can help you remember like what like a cytosine could look like would be something like what I drew over here, because there's a lot of uh hydrogen bonding that can happen here, right?
Yeah.
So, um, but what do you think? So cytosine deamination. Well, what do you think the deamination means?
Removing the NH2 group.
Exactly. So what do you think it would be replaced with?
I don't know what it's replaced with.
Hmm?
My thought was nothing, but I don't know. Uh, yeah, I don't know.
So, think of things that can then produce hydrogen bonding.
I would think of like an alcohol, like the like sort of like
What else could it be?
I was thinking of like an alcohol.
Hmm. Yeah. What else besides that?
Um, like a carbonyl.
Yep, like that.
Yeah.
Because, oh, I should have also said that deamination um means that you're going to be losing. So we're going to have ammonia as like a byproduct. Um, and what we're adding is essentially like water. So that's how you can get. So this is cytosine, and then this is uracil. Um, so yeah, just think of cytosine deamination. Like I would, I would try to memorize the structures for these, but yeah, if it says deamination, then you know that the cytosine or whatever they're talking about must have an amine in it, and you want to get rid of the amine, which means that you're going to have ammonia as a one of the products. But what is being like added in the mechanism for that, and it's going to be like water that's added um to give you the carbonyl oxygen, which can uh be an hydrogen bond acceptor,
Yep.
right? Because there's hydrogen bond um acceptors, there's hydrogen bond donors.
Yeah.
So yeah, so you want to think about what things can make something like. So what I mean by that really then is um so yeah, so let's say we have something like this, um like that. I mean, yeah, it's not perfect, I don't remember exactly, but you can see that. Or and then let's say we have our okay, so like, okay, so basically what I'm going to circle in green like this and these can be hydrogen bond acceptors. Oops. Wow. Whereas this can be a hydrogen bond donor. So you want to be thinking in terms of you want to be thinking about the pyrimidines, right? As having like either something like cytosine where you have these can act as hydrogen bond donors. This one can act as a hydrogen bond acceptor. And what it can like kind of like bond to would be something like this. So um so I would I would just make sure to look at the different structures for the different nucleotides and also like where certain things are, because you're pretty much just gonna have two types of things. You're gonna have like um so okay, if I just do this one more time, let's say this here, and this here. Um and then let's say I have something like this. Um so I just drew like I don't even think this is like an actual nucleotide that I just drew on the right, but what I want you to see is just that I'll use pink. So any amine will donate its hydrogen bond to a carbonyl oxygen, and we see that here and here.
Yep.
And a carbonyl oxygen will accept a hydrogen bond from the amine, right? So that can help you out. So like for the image of that I drew for cytosine here, right? The guanine would have to have some type of hydrogen bond here, let's say like HN, and then here with a double bonded oxygen. So something like this. I mean, like, with two rings, but just make sure that you I would say to look at to memorize the nucleotides, the bases that I mean, and understand how they're basically just going to be two things. There's going to be amines which can donate hydrogen bonds, and carbonyl oxygens which can accept hydrogen bonds. So you're going to want to think in terms of, is this going to be an amine group, or is this going to be a carbonyl oxygen group? So okay. So they're saying, what's the effect of cytosine deamination on the 5-terminal cytosine of the following DNA sequence? So just that one, I guess.
Yeah.
So yeah, what do you think it'll be?
It would be B.
Um, well, what else could it be? So, so the deamination would turn cytosine into uracil, right?
Yeah.
So this would be that, right?
A stop code.
Yeah. Mhm.
I see. Um, I just don't see how the others would affect the other options.
So
No,
if we introduce a premature stop codon, we're gonna get a shorter protein, right?
yeah,
And in size exclusion chromatography,
I see.
which things like would elute first?
Faster, bigger ones would lose faster.
Yeah. So if we're doing if we have a smaller one, it should elute slower.
Yeah, I see.
Yeah. So yeah, this was a good one because 30% chose B.
Yeah,
Hmm. But yeah, that was a good question because it incorporated a lot of other stuff.
yes.
But yeah, okay, next one.
Which of the following mutations will result in a frame shift mutation? Insertion or deletion of one nucleotide? I think so. Insertion or deletion of two codons. Deletion of three bases. Um. hmm. And it's I think just one.
Why not the others?
Has two codons. It's it's not influencing. I guess here it's like terminology, more of a thing. Like just like three bases
Yep,
would like be like one codon, if I'm not mistaken.
yep. So,
And
mhm,
sorry.
go on. Go on.
Um, and a codon is like, well, three bases. Yeah, I'm not sure where I'm
So
going, but yeah,
yeah, so a substitution won't change the reading frame, right?
right,
Insertion or deletion would, unless they're multiples of three, right? So
right,
if you insert or delete three bases, it's gonna maintain the reading frame. If you insert or delete two codons, that's six, bases, or nucleotides, uh, which is a multiple of three, so that's not going to change the reading frame either. But yeah, if you insert or delete just one nucleotide, yeah, that's gonna mess up the reading frame.
yeah,
Mhm.
yeah. I like that, thinking of it as like if it's a multiples of three
Yep.
of bases that are inserted.
Mm-hmm.
And then just remembering like codon has three bases.
Yep.
Uh,
Mhm.
yeah, yeah. I'm just trying to sink it in.
Mhm.
Solution of one nucleotide. Which of the following processes is most likely to result in a deleterious effect? Addition of a stop codon at the 3' end of the final RNA product. I don't think so. Introducing 14 nucleotides into the intron region. It's intron, so I don't think so. Deletion of a nucleotide near the 5' end. Addition of a nucleotide. Wait, deletion of a nucleotide near the 5' end and addition of a nucleotide near the 3' end. Probably. Let's see. And performing base pair excision in a location where purines from two different strands are interacting. And between C and D. Now let me think. Probably not C. D. Performing base pair excision in a location where purines from two different strands are interacting. So you're like eliminating bases. Where purines from two different strands are interacting. But it's not deleterious, is it? Like if the purines, two of them, usually don't you have like a purine and a pyrimidine interacting? So like if you do base pair excision, you're like repairing it. So I don't think that would be negative. I think it would be positive. So I think so the only one left would be C.
Yeah, because if the purines on two different strands are interacting, that's not good. That's not, you don't want that. So base pair excision is gonna remove that base, right? And put in so something will remove it. What would add the correct one?
Like DNA polymerase,
Uh-huh. And what would happen after? Like, is that it?
ligase.
Yep. Good.
Yeah.
Yep. Good job. So yeah, it seems like you have a good handle on this. And yeah, I would just say to memorize the structures of those of the purines and pyrimidines.
I have it here.
Yeah, there you go. So yeah, you could see how like here, here, or rather, well, I mean here, here. Well, wait, not that X. I don't really know what that X is, but like these should be like the three whatever hydrogen bonds there, and here, here, here would be the hydrogen bonds there.
Yeah, I'm not sure what that X is either. Maybe this is better, but
Uh,
I'm Mhm. R,
I think the X is just the thing that's bound to the ribose.
right?
So, um,
Oops, now I don't know what happened. I want to go back to that.
oh yeah, you can just go to images, go up. Yeah, right there. Yep.
But I really like that one. Can't find it, can
Oh, I saw it here. Wait, was that the one?
I? No. This one, maybe? Yeah, I think this one. Mhm.
Oh yeah, yeah. Yeah, the X is just going to be the thing that's bound to the ribose,
Try to look
right?
online, it's fine.
Yep. So, so yeah, yeah, so I would like, memorize these. So what I would do is like for the cytosine, like and guanine, I would just know that like, okay, that, and that probably participate in hydrogen bonding, and this, and this participate as well. And then for the adenine and thymine, right, we have like this and this, I guess. And then for the adenine, I guess we have this, and that. Um, but yeah, I guess this image here can kind of, yeah, shows you like those actual hydrogen bonds.
Yeah, do you want me to click on it?
Yep. So you can see here we have um for the thymine, right? We have the carbonyl oxygen that acts as a hydrogen bond acceptor, and then the amine here, which acts as a hydrogen bond donor. And here, yeah, like the NH, the amine doesn't have to always hydrogen bond with oxygen, right? Like, with FON, like those can hydrogen bond or form hydrogen bonds. So, yeah, that's what you have for the adenine and purine. And then for, sorry, adenine and thymine. And then for the cytosine, right, we have that amine up there, we have the nitrogen there, and the carbonyl oxygen, and then for the guanine, we have that stuff as well. And then I would just add to this, just, if you replace this with, a carbonyl oxygen, that's going to be your uracil. So yeah, I would just remember those, and yeah, I think you're good pretty much.
Yeah. I have to just draw these a couple of times.
Yep. Yep. Yeah. Practice with that. Um, and yeah, I think you're in a good place.
I try to internalize that.
Yeah.
Ask more questions.
Mm-hmm. Mm-hmm.
Yeah. So I guess this would be our last meeting.
Yeah. So yeah, you did a great job. You prepared with the really, really hard tests, and put in a lot of work, which is pretty incredible considering, you're working. So I think you're in a very good place for your test. Um, so yeah, so tomorrow just take it easy, like, do Anki and things like that, but don't do anything to, that don't do anything that will like tire you out or anything like that. And yeah, go to bed like a few hours early, because we talked about how, the day before your test or the night before your test, like you're gonna probably fall asleep like 2 hours later than when you go to bed, so you can go to bed earlier. Um, and yeah, I think you're gonna do an excellent job.
Right. Thank you.
Yep.
And thank you for helping me out these
Yeah, of
last,
course.
well,
It's been a
probably
pleasure.
more for sure, for a lot of weeks.
Mhm,
Yeah.
mhm. Yeah, it's been a pleasure. So yeah, so let me know. Yeah, let me know how it goes. And yeah, I think that you're gonna do a great job.
Fingers crossed.
All right.
Right.
So yeah, take care, get some rest. And then tomorrow do, you know, some Anki maybe, and go to bed early. And Saturday take your test. And after that, just don't think about it uh for a while. Like at least until, whatever, you get your grade back. So yeah, do you have like any like vacation or things like that you're gonna do after?
No. I think I'll go to New York for like the weekend after. So that's something.
Yeah. That's good. Yeah. Yeah, so I think you're gonna kill it. All right. So good luck, and yeah, let me know if yeah, you need anything else, but you're gonna do a great job.
All right. Thank you.
All right, take care, okay? Good luck and see ya.